Hello students, welcome to CCLMS. This is Dr. Praveen Dutt, working as a lecturer in zoology, Government Degree College, Mayilavaram. And in this class, we are going to learn about cloning vectors. This is the first part of cloning vectors. Learning objectives of this lesson. After this class, you learn about cloning vector, the foundational facts about the cloning vectors, and what are the characters of cloning vectors, and the different types of cloning vectors used in genetic engineering, and especially you learn more deep about the plasmids and the characters of the plasmids and different types of the plasmids. Besides, you will also study one of the type study that's called pBR322 and pUC plasmids in detail. You are going to learn about all these things in this lesson. So probably you might have heard about the word genetic engineering. In this era of biotechnology, genetic engineering is the most popular word. So what is genetic engineering? Genetic engineering is the process of joining two different DNAs to make a hybrid DNA. In this market today, you will see different kinds of genetically modified products. And this genetic engineering requires the two important genetic tools. The number one is enzymes, the DNA enzymes. The other two, the second one is cloning vectors. In the previous lessons, you learned about different types of restriction enzymes like restriction enzymes, DNA polymerases, DNA gyrases, DNA ligases. And these enzymes are all useful in genetic engineering. Well, that's first half. The remaining second part of the genetic engineering is cloning vectors, which we are going to learn in this class. So what are cloning vectors? Cloning vectors are the vehicles used to transfer the desired gene into a host cell. In cloning of genes or genetic engineering, we will select the required gene and we introduce that gene into the host cell. And these cloning vectors, they carry the desired genes into the host cell. It means the cloning vectors are DNA carriers. They are called DNA vehicles because they carry the DNA fragment into the host cells. And these cloning vectors are themselves are the molecules of DNA, and they are capable of self-replication. And let's see what are the ideal characters of an ideal vector. How to select an ideal vector? See, in gene cloning, we need to select a right vector for transfer of genes. So what are the characters of an ideal vector? The first of all, the vector, the cloning vector, the first quality is it must able to carry the DNA fragment. Should have ability to carry the DNA, number one, and it able to replicate in the host cell. And this should be easy to isolate from the host cell. And this must be small in size with a low molecular weight, should have an ability to self-replicate. And this ideal vector must have a multiple cloning sites with a selectable marker genes. So what are the characters of an ideal vector? See, in we have a different cloning vectors used widely in genetic engineering. Before select a cloning vector, that vector must have these following characters. The first one is every cloning vector must have an ability to carry the DNA. It should have ability to transfer the DNA into the host cell. That is the first and foremost character. And second important character is self-replication. It should be able to replicate on its own in the host cell. And the third is that cloning vector must have multiple cloning sites. You studied about restriction enzymes, restriction sites, right? A cloning vector must have a multiple restriction sites, and it should allow the multiple restriction enzymes to work on different kinds of restriction sites on it. And it should also carry some selectable marker genes. That cloning vector must carry selectable marker genes in it. And the fifth point is a cloning vector must have a site of origin site for the replication. If you are running a running race, there will be a starting point for the beginning of the race. Similarly, the cloning vector also having a starting point for replication. That's called the site of origin. So cloning vector must have a site for the replication, that is called origin site, which means the beginning site. And this cloning vector must be able to produce high number of copies, called high copy number. It is always advised to have a small sized vector. The cloning vector must be small in size, and if it is small in size, automatically it is low in molecular weight. Self-replication, multiple copies, as I already told you. And this cloning vector must be easy to isolate, easy to purify, and it should contain control elements. A cloning vector must contain control elements like promoter and operator. So these are the qualities for a cloning vector. So if you want to do cloning, the first and foremost thing is to select an ideal vector, the suitable cloning vector. So that suitable cloning vector must have all these characters. What are the uses of these cloning vectors? Generally, these cloning vectors are used in cloning experiments. For the cloning process, we use these cloning vectors. And to make genomic libraries, we need cloning vectors. And to prepare DNA probes, especially in case of DNA fingerprinting, we need DNA probes. For that purpose, we need cloning vectors. In many genetic engineering experiments, we need cloning vectors, especially vaccine production. Recently, we are suffering, the world is suffering with a pandemic called coronavirus. And we need to prepare a vaccine for coronavirus. How do we prepare a vaccine for coronavirus? You need to pick up the gene from the coronavirus, and you must introduce that gene into the host cell. Then how you will introduce this gene from the coronavirus into the host cell? For that purpose, we need vector called cloning vector. So for many uses are there for these cloning vectors. And let's see the types of cloning vectors. Majorly, there are 4 types of cloning vectors. Number 1 is plasmids. The second one is bacteriophages. And the third one is cosmids. Fourth one is artificial chromosomes. Here, artificial chromosomes are 2 types. BACs, B-A-C, bacterial artificial chromosome. Y-A-C means yeast artificial chromosome. Bacterial artificial chromosome and yeast artificial chromosome. And in this class, we study about plasmids. The first and foremost important cloning vectors are plasmids. What are plasmids? See here in this picture you will see a bacteria. In that bacteria, this is this brown colored ribbon-like structure is called DNA, the bacterial DNA. And this green color is cytoplasm. In the cytoplasm of the bacteria, besides the bacterial DNA, you can see the blue colored one. This one is also this one also a DNA, but this DNA molecule is circular in shape and it is also double stranded, and this one is called plasmid. The original chromosome is this one, the bacterial DNA, but this is also DNA molecule, extra. That is why it's called extra chromosomal. So plasmids are circular, double stranded, extra chromosomal, other than this original DNA, self-replicating DNA molecules. It means the plasmid also a DNA molecule which can self-replicate, present in the bacteria other than the bacterial DNA, and this one is called plasmids. And these plasmids are present in the cytoplasm of bacteria. There are many in number and they are different in sizes. They may vary from 20 to 40, sometimes 1,000 copies of plasmids are also present in a single bacteria. In this picture also you can see this structure of this is a bacteria cell. This is the original genome. Besides this circular, double-stranded, extra chromosomal structure, self-replicating DNA molecule is called plasmid. And next one is types of plasmids. Generally, these plasmids are divided into natural plasmids and artificial plasmids. See, the natural plasmids are the plasmids which occurs naturally in the bacteria. They are called natural plasmids. And the artificial plasmids they are man-made plasmids. They are manufactured or they are modified from natural plasmids. Natural plasmids are taken out and they are modified. It means some genes are added to the natural plasmids, some genes are deleted, and that natural plasmid is modified, and finally it was made an artificial plasmid. And these natural plasmids are available naturally from the bacteria and they are large in size, whereas artificial plasmids are small in size. And these natural plasmids do not contain any genetical markers, whereas the artificial plasmids contains genetical markers. And the unique restriction sites for common restriction enzymes were absent in natural plasmids, whereas they are present in artificial plasmids. And these natural plasmids are not suitable for genetic engineering. Artificial plasmids are suitable for genetic engineering because we can introduce the required genes into this artificial plasmid, and we can also remove unwanted genes from these artificial plasmids. That is the reason why artificial plasmids are useful in genetic engineering. They are most suitable in genetic engineering. And we have another type of classification of plasmids called conjugative plasmids and non-conjugative plasmids. Well, conjugation is a reproductive process in bacteria. Here you see the two bacteria and a conjugation takes place, fusion of DNA. So here you will see a separate structure called pilus, where the two bacteria meet through this pilus, where the plasmids are exchanged. And this process of exchanging DNA, and this process is called conjugation. Some plasmids have the genes for conjugation and they facilitate conjugation in bacteria, and that kind of plasmids are called conjugative plasmids. Whereas the genes codes for conjugation are absent in some plasmids, and such kind of plasmids are called non-conjugative plasmids. Conjugative plasmids, they contains genes for conjugation. What are those genes? They are called Tra genes. Here you can say Tra, T-R-A. That's the name of the gene which facilitate conjugations. And the Tra genes are present in conjugative plasmids. They are absent in non-conjugative plasmids. Conjugative plasmids are large in size, whereas non-conjugative plasmids are small in size. And the stringent control of DNA replication in conjugative plasmids, whereas relaxed control of DNA replication. Conjugative plasmids are low in number, whereas non-conjugative plasmids are high in number. And here is another classification of plasmids. Plasmids divided into three types here. The fertility plasmids. As I told you, these fertility plasmids contains genes for conjugation. The other name for conjugative plasmids is fertility plasmids. And the second one is resistance plasmids. Some of the plasmids, they contain genes for antibiotic resistance. Like there are different antibiotics like rifampicin, we have chloramphenicol, amoxicillin and tetracycline. Different types of antibiotics. Some plasmids contains genes in resistance to these antibiotics. Such kind of plasmids which contains genes for antibiotic resistance are called resistance plasmids. And finally, we have another plasmids called call plasmids. And these plasmids contains genes which produce a poisonous protein that can kill the bacterial cells, the neighboring bacterial cells. And such kind of plasmids which contains genes for harmful proteins are called call plasmids. And now let's have a look over nomenclature of plasmids. As you and I have names, plasmids also have names, right? So there are certain rules to be followed to name every plasmid. Every plasmid must be given a name. And that name, the first letter must begin with small p of English alphabet. That small p, p for plasmids. And after the small p, followed by the first letters of the names of the researchers. Whoever invents that plasmid, whoever manufactured that plasmid, the name of those scientists or the first letters of those scientists can be given in the second and third position, followed by a number. Every plasmid may have a number or may not have a number. This number is given by the researcher or the scientist or according to the number in the laboratory. So likewise, a name is given. The first letter is always P, small p. The second and third letters are with the first alphabets or first letters of the names of the researchers who invented this plasmid, and followed by a number given by the researcher. And now let's study pBR322. This is one of the plasmid. Here you can see these two are the names of a plasmid. pBR322 is the name of a plasmid. pUC is another plasmid. In this class, we will study these two plasmids in detail. So the first one is pBR322 plasmid. This is the most popular cloning vector for E. coli. So this pBR322 is the artificial plasmid. And this is the first artificial plasmid made in the world. This is the most popular plasmid used all over the world, widely by the many researchers, and hence this is called mother of all plasmids because it is so popular, widely used all over the world. And many of the plasmids are origined from this plasmid, hence it is called mother of all plasmid. It is a first artificial plasmid, and the name is pBR322. P denotes, as I told you, plasmid. B and R, these are invented by, or these were produced. This plasmid is made by two scientists called Bolivar and Rodriguez. So hence the first letter of the Bolivar, B, and the first letter of the another scientist called Rodriguez, called R, and these two were added after small p letter, as I told you earlier. And 322 is the clone number or the strain number given to this plasmid. So that is about pBR322, and it is having 4,361 base pairs or nitrogen base pairs are present. So pBR322 is a circular double-stranded DNA. Is a circular double-stranded DNA. And this pBR322 contains restriction sites. Nearly 500 plus restriction sites are present in pBR322. And nearly 66 restriction enzymes can cut this plasmid, can work on this plasmid. And this pBR322 contains two selectable antibiotic resistance genes. Here you can see this is the plasmid, circular double-stranded DNA molecule, this pBR322. And this pBR322 contains restriction sites for many restriction enzymes. These are the restriction enzymes: EcoRI, HIND3, BAMH1. These are all the different restriction enzymes. Different restriction sites are present. Likewise, around 528 restriction sites and 66 restriction enzymes can cut this plasmid. And the very important character of pBR322 is it is having two antibiotic resistance genes which are called selective markers present in this pBR322. The one is ampicillin resistance gene, that is called a BLA gene, and this BLA gene, which is in green color, shown green color here, it gives resistance for the antibiotic called ampicillin. Another gene called a TET gene, TET4 gene, means TET means tetracycline. And this region having a gene which gives resistance against an antibiotic called tetracycline. So these ampicillin resistance genes, tetracycline resistance genes, these two are the characteristic feature of pBR322, and hence they are called selective markers. Two selective markers are present, 4362 kilobase pairs. And every pBR322 plasmid must have origin of replication. This is a red colored one is called, this is a starting site for replication. And this is called ORI. This is called ORI site. Replication site called ORI. It means this is the place where the replication begins. This plasmid starts replication, and that replication begins from this place called origin of replication. It is called ORI site. And this pBR322 contains restriction sites for many restriction enzymes like EcoRI, HIND3. These are the restriction enzymes, and these are the restriction sites present in this plasmid. And let's see the advantages and disadvantages of pBR322. First, let's see the advantages. This is very small in size, so that its molecular weight is very less. It is very easy to isolate. It is very easy to purify these plasmids because of its low molecular weight. And the presence of two selectable markers, that is AMP gene, TET4 gene, that is antibiotic resistance genes, two different genes we have, that is ampicillin resistance gene as a marker and tetracycline another marker. Two selectable markers are present, and this pBR322 can be amplified up to 3000. It means up to 3000 copies it can produce. It also have some advantages. Let's see. And it has very high mobility. Because of its low molecular weight, it is very highly mobile. It means it can enter into any kind of cell very quickly, very fastly. It can enter into any bacterial cell. That's called high mobility. And it cannot carry DNA fragments more than the length of 15 kb. It means the pBR322, it carries genes and introduced in a host cell, but the length of that genes it carry must be below 15 kb. It means if the length of the DNA is more than 15 kb, the pBR322 cannot carry that DNA molecule. That is the disadvantage. Now let's see another plasmid called PUC plasmid. PUC. P denotes plasmid. The first letter is P denotes plasmid. Here UC stands for University of California. Actually, this plasmid was constructed by Messings in 1983 and his friends. And all people belong to University of California in America. Actually, they have a right to give any name. Generally, I told you the second and third letter must be the name of the scientists who invented this or who constructed this plasmid. But here, this Messings, the one who constructed this plasmid, he gave the name after his university. That is, he is working in University of California. So he named this plasmid after the name of the university. So PUC means plasmid of University of California. It was constructed in that university. And this is the source for, actually this PUC plasmid is modified from pBR322. pBR322 plasmid is modified into PUC plasmid. And in this PUC plasmid there are many modifications. As a result, a number of plasmids are produced in PUC series. That's called a series. Likewise, PUC 8, PUC 9, PUC 12, PUC 13, PUC 18, PUC different types. You know, if you see a movie called Bahubali 1, Bahubali 2, Bahubali 3, like that, a series. Likewise, PUC plasmid also have a number of series continually from 8, 9, 10, 11, 13 up to so many number of PUC series. That's why we name it as PUC series. You know, it is constructed from pBR322 and it is having 2676 base pairs. 2676 base pairs. And this is the PUC plasmid. As it is developed from pBR322, all the characters of pBR322, you can see those characters here. See, in pBR322, you have origin site, they have ORI site, right? Similarly, here also ORI site, right? It is having ampicillin resistance gene. That ampicillin resistant gene also present here. Then what is the specialty of PUC plasmid? Means, in addition to this genes of pBR322, it is additionally having a LAC gene. LAC operon gene called LAC jet gene is present here. Additionally, this gene was added and hence this is modified. So PUC plasmid specialty is to have additional gene called LAC jet gene, LAC operon gene called LAC jet gene. Like pBR322, it is having restriction sites for many restriction enzymes like BAM, H1, EcoRI, HIND3. Likewise, you can see these are all the different restriction enzymes and the restriction sites. Similar like pBR322. So that is about PUC plasmid. And let's conclude this lesson. In this class, we learned what are cloning vectors. Cloning vectors are the vehicles that are used to transfer the desired genes into the host cell. There are different types of cloning vectors, maybe plasmids, bacteriophages, cosmids, and artificial chromosomes like BACs and YACs, bacterial artificial chromosome, yeast artificial chromosome. And we studied about plasmids. Plasmids are the circular, double-stranded, extra-chromosomal, self-replicating DNA molecules present in bacteria. This is the most popular cloning vector used in genetic engineering or plasmids. There are many varieties of plasmids, out of which we studied pBR322, which is the first artificial plasmid, which is widely all over the world called the mother of all plasmids, and we studied its structure. And we also studied the PUC plasmid, and we studied the uses of cloning vectors in gene cloning and DNA library making, genetic engineering, vaccine production. Likewise, there are many uses for cloning vectors. And in the coming classes, we are going to learn the remaining vectors in the next class. Thank you very much for watching this.